Hi, welcome back to educator.com. This is the lesson on vision. Before we talk about the eyes themselves, we need to look at the accessory structures that are around the eye, that have to do with the eye, protecting them, allowing them to function properly. First, the eyebrows. These are one of those areas that we've retained a lot of thick terminal hairs from our mammalian ancestors. Of course, they're really concentrated on the top of the head, but if you ever wondered why do we have a lot of hair still right here, part of the reason is just expression. Being able to, see someone's eyebrow movements with respect to their facial expressions is important for the socialization that humans have. But beyond that, in terms of protecting the eye, when you sweat a lot above the brow, the sweat will tend to get caught in these hairs and prevent a lot of it from coming into your eyes. Now, if you sweat a lot, you're not going to be able to prevent all of it, but that's a nice function of eyebrows. The eyelids, the technical term being the palpebrae, they're made up of skin, muscles, a very, very, very tiny bit of fat. But you could think of those as like windshield wipers. Every time you blink, they get a little bit of fluid over the surface of the eye, helping to clean the eye, lubricate the eye, protect the eye. And of course, you have that automatic reaction where you blink if something comes to your face really quickly. So that's important in terms of protecting the surface of the eye. Eyelashes also protect the eye. They prevent a lot of dirt, microorganisms, little bugs that we don't notice from coming into the eye itself. Eyelash length is mainly genetics, unless you're, using some kind of product to accentuate them. An interesting thing about eyelashes is the tarsal glands. The tarsal glands are a modified kind of sebaceous gland. Now, sebaceous glands are associated with hairs in general, but the modification of the tarsal gland makes it so that there's even more of a lipidy kind of fatty secretion that comes out with the eyelashes, and not only kind of protects them, but it also makes it so that they don't clump together. It helps keep them separated. So those tarsal glands associated with each little eyelash you have, sometimes they can get infected. Sometimes a little bit of bacteria, dirt can get into them. They can get inflamed. And as a result of that inflammation, you can get what's called a stye. So if you ever heard somebody say that they had a stye on their eye, it's because of the tarsal glands getting inflamed. Skeletal muscles. You have 6 extrinsic muscles attached to the outside of the eye. On the next slide, you're actually going to be able to see those a little bit better. You can't see them just by looking at someone's face, but behind the eyelids, attached to the white part of the eye, you have all of these skeletal muscles. And of course, they allow you to move your eyes all over the place. The conjunctiva is a layer that's around the surface of the eye, and it also curves up on the underside of the eyelids. It's made up of a lot of epithelium that's meant to protect the eye. It's just another form of protection. And when it's healthy, when it's not infected, you don't really notice it. But when you get conjunctivitis, the infection of the conjunctiva, also called pink eye, it's very noticeable. And because of that infection, you get dilation of all the blood vessels. You get swelling in that area. And it can be very irritating, very annoying. And that happens because of, just could be irritation of that layer of that tissue. It could be a bacteria or virus that's making that happen. And generally, it's something that's only going to last several days. The lacrimal glands. If you remember from the skeletal lessons, there's a lacrimal bone, and it comes from a Latin word that has to do with tears, like lagrima in Spanish. So the lacrimal glands, they are those tear producers. The lacrimal gland itself is about the size and shape of an almond, and they are located in this region of the eye. You're producing about 1 milliliter of tears per day. Of course, you can go beyond that if there's a lot of crying going on, but even when you go the whole day without shedding a tear, you're still producing tears. They're just not exiting the eye. Every time you blink, you get a little bit of that fluid going over the eyeball. The nasolacrimal duct is what leads those tears out of this area and through the lacrimal bone and part of the maxillary bone into the nasal conchae, the nasal cavity. You have this thing that's nicknamed a lacrimal lake with each eye, and it's kind of like a space for excess tears to hang out in. If that lacrimal lake gets overflowed with tears, they're going to come out the eye. So the act of crying, you're producing so much of those tears that that lake overflows. Your nasal lacrimal duct isn't able to drain all of it out in those moments, so they're going to come out the eye, down the cheek, etc. The duct, since it takes tears down into the nasal conchae area, that's why when you are crying a lot, you're going to get the, like you're having the sniffles at the same time, because you have a lot of tears going into that region. And besides all of these, you've got orbital fat. The orbit of the eye is formed by several bones that make that, classic looking eye socket. And cushioning the eye all around the exterior, attached to the white part, known as the sclera, you have a lot of fat. And it's meant to cushion. It makes sense that you want that cushioning around the eye because you have some very hard bones that are right next to that eye. So the orbital fat, nice cushioning. We're going to talk about the eye in terms of 3 tunics, and we're going to start with the outer tunic, this outer layer, go to the middle, and then to the inner. So the outer fibrous tunic is the other nickname for it, is incredibly fibrous. It's made up of a lot of connective tissue fibers, mainly collagen and elastin. The white part of the eye is called the sclera. Very hard. It varies in terms of the thickness and hardness depending on what part you're at. If you go to the back part of the eye, and this is a superior view of the right eye, in the back next to where the optic nerve is, it's actually going to be a bit harder. It gets a little less hard when you get closer to the front or anterior part of the eye, but in general, the sclera is very tough. There's a lot of science fiction movies or tales of, someone stepping on an eyeball and it immediately squashing as if it's like a balloon or something. It's not that soft. Yes, with enough force on an eye, you're going to collapse it, but it is very tough. And if you've ever dissected a cow eye or sheep eye, you know that. The sclera, very tough. You have attachment for all the muscles. You can see all those extrinsic muscles, most of them here. Some of the others you can't see because they're on the underside, the inferior side. But besides this part of the outer tunic, which the majority of which is sclera, you have the cornea. So the most anterior frontal portion, the part that you put a contact lens on, that's known as the cornea. And it's made up of some squamous epithelium, about 5 to 7 layers thick, and it is transparent. At least it's supposed to be transparent. And that's important because you don't want to disrupt the rays of light that are coming through there. That is the first layer, that is the window of the eye in which light first passes through. So you don't want there to be anything getting in the way of light, because then that would change your perception of what's outside of your head. Just beyond that cornea, and it's actually hard to the touch, it's pretty tough, just posterior to it is a little cavity called the aqueous humor, and a lot of textbooks call that the anterior cavity because it's the most frontal space just behind the cornea. And there is fluid in there, and that should also be clear. If you look very close at someone's eye, directly on the side of it, you can see that little aqueous humor just behind the cornea. You produce fluid there, and it's supposed to drain through what's called the canal of Schlemm. I love that term. Schlemm is spelled like this. The canal of Schlemm actually drains excess fluid out of the aqueous humor. If that gets backed up and there's too much pressure, it could actually lead to something called glaucoma, which I'm going to tell you more about later. But the interesting thing about the cornea and aqueous humor is the cornea is one of those few places in the body, other than the outer parts of the epidermis, where there are no blood vessels directly adjacent to it. So how do the cells of the cornea get nourished? Well, it's through the aqueous humor. So you have little bits of gases and nutrients coming into that fluid cavity, and since that's directly adjacent to it, that's what's nourishing the cornea. the cornea can get scratched, it can get damaged. With enough impact, it could be completely destroyed. And actually, one of the most common transplants done to human beings is corneal transplants. And you can actually get corneas from cadavers, from dead bodies, after death. The majority of organs you need to take out of a donor while their heart is still beating, while the cells are still getting oxygenated and getting nourishment. But one of those tissues you can take several hours after death is the corneas. And so corneal transplants, typically you get them from cadavers. When we go deeper, you have a lot going on in the middle tunic, also called the vascular tunic. Vascular means having to do with blood transport, getting blood into an area. So there's a lot of circulation coming into the eye, and it's thanks to the middle tunic. So the choroid, I'm going to color this in red. And it is red in a living eye. If you dissect a cow eye or sheep eye, the choroid's going to look actually black because once you don't have all the oxygen and actually living blood cells going into there, it's no longer red. It's very faint, but this little layer, you can see it, is right in between the sclera, which is here, and the retina, which is actually part of that inner tunic. So here's the choroid in red. And if you follow this all the way to the anterior portion, you can see how it connects to these parts called the ciliary bodies. And I'll cover those momentarily. The choroid is mainly for blood flow. And it also has a lot of pigment. There's also melanin in there. And the purpose of that is when light enters the eye, you don't want it to stick around. You want light to enter the eye, be noticed by the retina on the back, and not be bouncing around. You don't want to be seeing something for a second or two after you've seen it. You want to get new light coming in and then going away. And one of the ways that you can absorb light and get rid of it once it's been first absorbed by the retina is by having a very dark interior that's going to absorb. So having that pigment added to this blood layer is able to absorb light as it comes into the eye. And like I said, it is providing nourishment to the parts of the eye that need it. Here's the iris. The iris is made of smooth muscles, actually 2 layers of smooth muscles. And they're called smooth muscles, not skeletal muscles, because, well, they're not attached to your skeleton and you do not have conscious control over them. They change based on how the light is outside of your face. If there's a lot of light, you don't need as much of the light coming into the eye. So you're going to have the muscles get more relaxed and not be constricted. And so that's going to actually make the iris bigger, which makes the pupil smaller. So the pupil is just a hole. I'm not going to draw on it because it's really, the pupil's not something that contains matter. It's not something that has substance. It's just a hole, and the hole is inside of the iris. If you're wondering why the iris looks like this, it's not round, well, this is a cross-section. It's actually a transverse horizontal cross-section through the left eye. And the reason why I know that is because of the angle of the optic nerve, which we're going to hear more about later. But the iris, made of smooth muscles, when there's not enough light, you want to actually get the iris expanded, constrict some of those muscles to make the pupil bigger. You make the hole bigger, so the little bit of light that there is available, you want as much of it as possible coming in. And of course, the pupil size is connected to the autonomic branch of the nervous system, the sympathetic and parasympathetic side, which you can review in some of those nervous system lessons. So the iris, let's make it blue. I have blue eyes. Smooth muscle, unconscious control. The pupil, like I said, that black dot, it's just a hole. And the reason why it looks black is because the inside of the eye is dark. The lens, I'm going to color in yellow. Now the lens should not actually be yellow. It should be clear. And the lens is very tough. The lens is meant to last a lifetime. It's very tough, made of crystalline fibers. And if you actually cut it apart, it almost looks like an onion, like it has all these different layers of these crystalline proteins. And it's meant to be clear. It's supposed to be clear because, like I said, when light's coming through the cornea, which is clear, through the aqueous humor, which is clear, and through the lens, which is clear, that's good. You don't want the light to be influenced or anything being added to it as it's coming in. So you want a clear lens. If your lens gets a buildup of calcified compounds, other molecules, it can make it so where it's cloudy. And we're going to look at a picture that has to do with cataracts later. Cataracts is the buildup of usually calcified products inside the lens, and it makes your vision kind of cloudy because you got this stuff in the way of the light coming through the eye. And something that you can do to get rid of that is cataract surgery. They take the lens out and they put a new lens back in. The lens is what changes in size to focus near or far. So your ability to look at something very far away, something very close up, that has to do with the lens. The ciliary bodies, which have little ciliary muscles in them, have to do with changing the lens shape. So attached to these ciliary bodies, and it's actually circular around the entire lens, because remember, this is a cross section, you have suspensory ligaments. Let's do that in green. So the suspensory ligaments right here. And those are all along the exterior part of the lens as well. If those suspensory ligaments pull on the lens, it makes it flatter. It actually makes it so that you're going to see farther away. When the suspensory ligaments kind of let go on the lens, it makes it more bulging, and that's what allows you to read something close, look at something very close to your face. So those are the basics of that middle or vascular tunic. Next up, the neural tunic, the most inner part of it. And it's neural because all of this has to do with sensing the light that's coming in and getting those signals to the brain. The most important part of this would be the retina. The retina is really what allows you to see. Now, of course, all those other parts I mentioned before, very important. But all those other parts could be working perfect. Nothing wrong with them. But if the retina ain't working or the retina falls off the back of your eye, it doesn't matter how well your occipital lobe is working, the lens, the cornea, etc. The retina is kind of this drape, curtain in the back of the eye that has all the photoreceptors, those cells that actually respond to those different wavelengths of light coming in. Photoreceptors, that's the next point. These are specialized cells all along the retina. You have about 130 million photoreceptors per eye. Way more rods than cones, and we'll discuss the differences between rods and cones in a little bit. But those two kinds of photoreceptors scattered all along the back of the eye. And depending on what part of the retina we're looking at, the amount of rods and cones can vary. The macula lutea is the full name of it, is an area right back here. And in the very center of this region is a point called the fovea. The fovea centralis is the full name for it. And the fovea is where you want light rays coming into the eye to be perfectly projected on. You want those light rays converging to that point. And if light rays are not coming on that point, you're not going to see clearly. So the reason why I am wearing glasses is because when I don't have my glasses on, the light rays from looking towards you, they don't quite come on that point perfectly. They actually come to a point, converge in front of it. When I put this lens in front of my eye, it changes the diffraction. It changes the angle at which light rays come in and makes it so that they come right there. And that's nice. I like being able to see clearly. So the fovea centralis is that part of the eye that you want light rays to be focused on. And we'll talk more about vision and nearsightedness versus farsightedness in a bit. So the macula, or the fovea, right there. The optic disc is a part of the retina that's right on top of the optic nerve. So notice that the optic nerve is slightly off to the side. So if we're looking down on the left eye, which we are in this case, the right eye would actually have an optic nerve coming more medial as well. So we're going to look at this in a little bit, how the optic nerves come kind of together and there's a little crisscrossing going on. So with each eye, there is this area right on top of the optic nerve where there are no photoreceptors. It's a blind spot. Now, you don't notice the blind spot of the eyes when both eyes are open, because if I consider light rays bouncing off of the object over there, coming into my eye right on where my optic disc would be on my right eye, the reason why I don't notice that, I don't notice it, which sounds contradictory, but the reason why I don't notice the blind spot is because my left eye is able to perceive what's there. So my left eye kind of allows sort of a cover up of the blind spot of the right eye and vice versa. Now, when you close one eye or the other eye, you're able to notice that blind spot. And we're going to do a test in a little bit so you can see that certain things might disappear when they're actually hitting just that spot of the retina. So there are no photoreceptors on that part, but photoreceptors all along the other parts of this green retina that I've colored in. The vitreous humor, in this case they call it the vitreous gel, is a gel-like sac that is taking up all of that space that's just deep to the retina and just deep to the lens. It's supposed to be clear as well. It's in the pathway of light rays, so you want it to be clear, but it's a gel that's meant to give the eye some integrity, to give it the right amount of pressure so that it's not collapsing, and it's a nice medium for light rays to go through on their way to the retina. If there's too much pressure in there, it could result in something like glaucoma. And if you've ever wondered, what are those little floaters that you sometimes see? You'll be looking at something and then it looks like there's almost this like little white dot that's kind of drifting along, and you blink and it's still there. What is that thing? Well, floaters can be little bits of cells that come off from parts of the eye tissues, from parts of the inner tunic, and they end up floating along in the vitreous humor, this gel sac. And eventually they go away. Eventually, you have white blood cells in your eye that eat up stuff that doesn't belong there. But it's kind of funny, those floaters. So that's how they occur. The optic nerve, you have one with each eye. All those different neurons associated with the retina, with those photoreceptors, come together. They converge in the optic nerve and come out the back of the eye, move medially, and then end up going through the thalamus and eventually to the occipital lobes at the back of the brain. So here's that blind spot demonstration. Here's how you do it. And you could switch. You don't have to close your left eye. You could close your right eye. But if you close your left eye and stare at the star with your right eye, make sure that the star is right in your center of vision. So you don't want to have your right eye looking straight at this black dot, because the black dot is what's going to be disappearing. So make sure that your right eye is right in front of that star, and start with your head close to the screen. And with that left eye closed, gradually move your head back, keeping your right eye looking at that star, gradually, slowly move your head back, and then at a certain point, that black dot will vanish. Now, the reason why some people it doesn't work for them right away is sometimes they're moving their head too fast, so they move their head so fast that they don't notice that disappearance. Because keep in mind, once you move past, just barely past the point where that black dot, the light rays from it are not hitting that blind spot anymore, well, then you can see it again. So try this a few times if it doesn't work, but eventually it should work for you. If after trying this 10 times it doesn't work, there's a lot of optical illusion books out there. There's other things you can look up online, but it's quite fascinating. So when the light rays bouncing off this black disc hit your optic disc, that's the blind spot. But having both eyes open, this demonstration would not work, like I explained before. So the lens function, the ability of the lens to change shape so that you can focus near versus far is called accommodation. Some people's accommodations just aren't enough to cut it. Mine, as I got older, like once I got to high school, my lens wasn't cutting it. And actually, my lens was kind of, since it's not doing enough to focus the light rays at the back of the eye, an eye doctor would say that my eye is too long. And if that doesn't make sense, I'll explain something in a little bit to help with that. But the reason why they say my eye is too long is because no matter how much my lens gets flat to look far, far away, it just can't get those light rays all the way back to my retina. So if my eye was a little bit shorter, then I would have clear vision. And it's the opposite with being farsighted. Those people's eyes are, in a sense, too short for what the lens is doing. So as the lens gets more concave, meaning the opposite of bulging, so a concave lens, a biconcave lens would look like this. Okay, that's what concave is. Now your lens can't literally flip itself like that, but what this means is if this is convex, getting more bulging, the more flat it gets, closer to this kind of inverse lens. You can call that getting concave. So getting concave means more flat. And that's what happens to focus on images from far away. The lens getting more convex, as in this particular image right here, is for images close by, close to your face. So for an image to appear clearly to the viewer, the image must be precisely projected on the fovea, which is right there and right there. All right, accommodation problems. This is why people need glasses or contact lenses. The term emetropia means proper vision. So if you have 20/20 vision, you don't have an accommodation problem. If you ever wondered about the 20/20 thing and why some people are 20/15 or 20/10, don't think that 20/20 vision is perfect. That's normal vision. That's what you want to have. That's expected. Here's how it works. This is a good way to explain it. 20/20 vision means that somebody with normal vision from 20 feet away can read a sign, read all the words on it properly, and a person next to them, also from 20 feet away, can read that same image, those same words perfect. So 20/15 means this, that a person with normal vision actually has to be 15 feet away. They have to move 5 feet closer to see that image. But the person with the 20/15 vision, they can still be 20 feet away and see it great, see it perfectly crystal clear. 20/10 is even more exaggerated. That person with normal vision, great vision, they have to be 10 feet away from those words to read it. And that other person with 20/10 vision, they can be 20 feet away. So yeah, you could think of 20/15 or 20/10 as better than perfect. But the term perfect vision, when we look at other species, there are birds out there, predatory birds, that have vision better than any human on Earth. So the term perfect vision is relative. The opposite is true with a relative of mine. I have a relative who had 2200 vision. 2200 vision means that a person with normal vision could read a sign from 200 feet away, but she had to be 20 feet away from it to read it. After getting LASIK surgery, that was corrected. And LASIK surgery involved actually lasering off parts of the cornea, making the cornea more flat to kind of assist the lens with the lens's incapability to get even more flatter. So the LASIKs made it so that now she actually has close to 20/20 vision. The only problem with that is now she needs reading glasses. So when you make the cornea a little bit flatter to assist with seeing in the distance, that ends up being a problem when you're trying to read something really close to your face. But at least you have that far-sightedness. So back to this. Myopia. This demonstration here shows how to fix myopia. That's being nearsighted. So I am myopic. I'm able to read things right in front of my face, but without my glasses, seeing things in the distance is quite a problem. So like I said before with the previous slides, that means that my lens just can't get flat enough to look at something far away. And as a result, the images are converging right here in front of or anterior to the fovea. When you put a lens like this in front of your face, a convex lens, or sorry, my bad, concave lens. The concave lens makes it so that you're assisting the lens with making those light rays hit the fovea. Now, you could kind of imagine how a lens inside of eyeglasses could be shaped like this, but I've often heard the question, well, what about contact lenses? When you put a contact lens on the surface of your cornea, it's not shaped like that. You couldn't even close your eyelid over that. It would hurt. That's true. The contact lens is shaped like this from the side. But the way they get the lens to behave like this is by playing with the densities within the material inside the contact lens. So if you change the densities with respect to the middle part versus the outer part, that's going to give it this property. And the opposite is true of hyperopia. Hyperopia means that you are farsighted. You actually see stuff in the distance quite well, but you have problems with up close. And that's not as common as myopia. But hyperopia is the exact opposite. Imagine having the inability to make your lens bulging enough to focus in on the light rays coming from right here. That makes it so that when you're hyperopic, the light rays actually want to go past the retina, which is not physically possible. They're not going to be able to go past the retina. But when you put a lens in front of somebody who's hyperopic, a lens that actually happens to be convex, then you're making it so that it hits right here. And the intensity of how convex or how concave it is has to do with how bad your vision is. So somebody who's even more myopic than me, like the relative I told you about, maybe that stuff was converging even closer to the lens. So yeah. All right, when we look at the retina, all these photoreceptors in the retina that are actually sensing the light and reacting to it and then sending the signal to the brain, we have two main varieties, rods and cones. So the rods and cones have similar structures, but very different roles. Here's how I remember it. Rods have to do with light and dark, but cones is color. So imagine color with a C, cones with a C. C for color, cones color. Rods is just the ability to see light versus dark in general. So the rods, these modified neurons we call rods, not very good at distinguishing a wide range of colors. And when the light's dim, you've probably noticed that when it's darker in a room, it is really hard to distinguish colors, more so than when it's light out. You know, looking at objects in the dark, you can tell that certain things are there, but in terms of, you know, red versus orange, very hard to distinguish, or blue versus green. So the cones are meant to allow us to distinguish Roy G. Biv, red, orange, yellow, green, blue, indigo, violet, the colors of the rainbow. So this is in the visual spectrum. There are three main kinds of cones. It's actually very similar to those old projection TVs. If you've ever looked at those old projection TVs that have those three different colors projecting up, you've got the red, the blue, and the green light. And coincidentally, there are red cones, blue cones, and green cones. And actually the majority, or most of the cones are red. But all three are there, and depending on what combinations of those are stimulated, that actually gives you a sense of all the different colors of the rainbow. When we look at the retina, there actually are no rods on the macula, on the fovea section. It's just a bunch of cones concentrated there. And so that's why when we look at something straight on, when we're focused on something, it's easy to see the detail, the rich colors there, because cones are enabling you to see kind of that clear, very bright image. Whereas on the periphery, where you have more rods, you can tell things are there, you can tell the colors are there, but wherever we want to observe something's shape, texture, color, we're looking straight on. When we look at the different rods and cones, this is actually a rod. The main difference structurally between a rod and a cone is actually this. If we look at this part here, you can see that it's kind of like a rod, kind of like a pole. The outer segment of a cone is shaped like this. And you still would have these little lines, these little discs, which I'm going to talk about momentarily. But imagine having another one of these right next door, but instead of having the outer segment be long, it's kind of triangular. But structurally, other than that, they're very similar. Bipolar cells is a kind of neuron that looks like this. So here are dendrites, axon cell body, axon, and here are the axon terminals. So receiving end, the sending out end. Bipolar cells are what are going to be right next to the rods and cones. So if you think about the retina as having a few different layers of cells, the light first hits some other cells before actually entering the rods and cones portion. So the photons stimulate bipolar cells, which then send the signal to the rods and cones. And depending on how the photons hit the rods and cones, that's how you see all the different colors and different images. The inner segment, just a little bit more superficial. It's like we're getting farther back in the retina, closer to the different tunics, closer to the middle tunic, closer to the outer tunic. So you got a lot of mitochondria in here, and then the area that's really doing most of the work in terms of actually giving you sight is the outer segment. So in the outer segment are all these little discs. These little discs just stacked on top of one another. And these discs are made of a couple different parts. We tend to concentrate on the rhodopsin. So rhodopsin actually is made up of two parts. Opsin is a protein, and then there's a pigment called retinol. And retinol is made of vitamin A, or it's made from vitamin A. So that's why they say, like, eat your carrots. Carrots, compared to other veggies and fruits, have a lot of vitamin A. And it's not necessarily true to say, like, oh, your vision's bad, eat carrots and you won't need glasses. No, eating carrots is allowing you to maintain your eye health. If you get a deficiency in vitamin A, it can lead to some problems over time. But yeah, without vitamin A, you're not going to be able to make that pigment retinol. And opsin is a protein. Depending on the structure of opsin, that determines how it's responding to Roy G. Biv, the red versus the violet versus the green, etc. So you're going to see different kinds of opsin within rhodopsin in the cones and the rods. The pigment epithelium is just outside of that. You're getting a little bit closer to those optic tracks that are going to be taking the signal through the optic nerve. The pigment epithelium, it does have pigment in it. There's a little bit of melanin in those regions, and there's the ability in those cells to actually eat up, sort of swallow up the older discs. So you can actually make new discs, these optic discs within the outer segments throughout your whole life. Now you can make these new discs, but if you damage the cell as a whole, if you destroy this cell, it's going bye-bye. That's why if you stare at the sun, if you're looking at extremely bright lights, that's way too much light rays. That's way too much energy coming into the eye, and it's going to fry these cells. So yeah, staring at the sun could definitely make you blind. And I have read some research suggesting that in the near future, we will be able to regenerate retinas. But as it stands now, once you lose your retina, you're not getting one back. But in the future, I'm confident we will be able to make retinas from stem cells within your body. When we look at the visual pathways to the occipital lobe, it's important to realize that we have stereoscopic vision. It's like having vision in stereo. So if this is the visual field of my right eye, this is the visual field of my left eye, and they overlap. And there's a few reasons why that's important. Number one, if I close my left eye and I look around with my right eye, I can see that my nose is blocking a lot of stuff over there. The orbit of my eye is doing a lot to block what's over on the left side here. But when both my eyes are open, I don't really notice my nose as much. I can see it's still there, but it's not as obvious to me because the parts that were being blocked by my nose and part of the orbit of my eye when I had my right eye open, when my left eye is also open, I can see those parts clearly, what was over here. Along with that, having stereoscopic vision or vision in stereo is good for depth perception. So because the eyes on humans and primates in general are in the front, we are able to see ever so slightly around the sides of objects, and we can tell how far something is in front of our face. Unlike other animals that have eyes on the sides, having eyes in the front as primates is important because if you consider our ancestors that were all arboreal, all in the trees from millions of years ago, swinging through the trees, having depth perception is important so that you don't run into a tree or miss that branch and fall and get eaten by something. So it's important in terms of evolution and us being successful. And even though now we're not in trees anymore, as with most apes are not hanging out in trees, having depth perception has a lot of importance. Just think about driving a car. I mean, without depth perception, there'd be a lot more accidents. So stereoscopic vision, having vision in stereo, you can see that the visual fields do overlap. Here's a little depiction of those visual fields overlapping. When light rays come in, here's the cornea, light rays, I'm going to depict them in yellow. The light that's coming from the right side of your face into the right eye is actually going to end up hitting the left side of the retina. It's going to end up coming in on hitting the medial side of the retina, and vice versa here. Now, the funny thing about that is, let's say we went back into the head, where these neurons actually end up crisscrossing. And we'll get to those terms in a sec, but if for some reason there was trauma or an infection that harmed these inner parts, let's say that this got completely severed where these blue tracks overlap. Think about how this is laid out. The blue parts are sensing light from the outer part. So even though if this part of this little chiasm, this little space in here, if that got damaged in the middle, it would actually cut out everything around the outside of your visual field. So it'd be like just blackness. You wouldn't be able to see anything out here, and you'd have a much thinner or less wide field of vision. And you can see that there is some crisscrossing going on. The red parts of the retina it's being depicted here as red. Don't think that this corresponds to cones or something, but they're depicting this part as red. You can see that the red parts kind of stick on the outside, the more lateral parts, and the optic nerves contain these little tracks. So you can see that this little black tube-looking thing, that's the nerve. And as I mentioned before, the optic nerves come out from the posterior part of the sclera, of the outer tunic, and they come in medially. They come in towards the center of your head, and there's some crisscrossing going on before it ends up at the occipital lobes, which allow you to see. So those are the optic nerves in green. The optic chiasm, and this is actually plural, chiasma, the chiasms right here, that's where you have the little crisscrossing of some of those optic tracks. So the optic tracks, as you can see right here, optic tract, the red and blue lines, those are all the tracks of neurons within the optic nerves that end up going back to the occipital lobe. So the occipital lobe is all of this here. And you can see that that's the final destination for these tracks. And remember, if you saw the nervous system lessons, the one on the brain specifically, there's the visual cortex that allows you just to be able to see, and then there's that association part that allows you to make sense of what you're seeing. But this is how the visual pathways get from the eye to the occipital lobe. When we look at vision disorders or conditions that affect the eye, I already mentioned myopia and hyperopia. Remember, myopia is nearsightedness, and hyperopia is farsightedness. So I am myopic. I've got this concave lens that's allowing me to actually see in the distance, and it's vice versa with hyperopia. Cataracts, that's what this picture is depicting here. If you shine a light in this person's eye, you can see that through the pupil, it's quite cloudy back here. Because remember, just posterior to the pupil, to that opening, is the lens, and the lens should be clear. Buildup of calcified compounds is going to make it cloudy, and cataract surgeries are very common, especially in older people. You can have a cataract problem as a younger individual. That would be not that common, but an injury could actually cause cataracts to occur. But yes, senile cataracts in more older people are more common. And like I said before, they will typically remove the lens and put in a new one. Very simple surgery these days. Glaucoma is an increase in the intraocular pressure, intraocular is a better term, inside of the eye. Where the vitreous humor is, if you have too much pressure in there, it's going to be pressing on a lot of the delicate parts within the eye that allow you to see. So not only is it painful, but it can make you blind. One of the causes is, I think I mentioned this earlier, if the aqueous humor in that anterior cavity gets too much. You have too much of that fluid production there. It's not draining through the canal of Schlemm. That buildup there can affect the pressure within the entire eye, and glaucoma, painful, can result in blindness. Glaucoma, you've probably heard that one of the treatments that you could use for it is use of cannabis, marijuana. My good friend from college, who's an eye doctor now, has told me before that the amount of marijuana a person would have to smoke to help with glaucoma is absolutely ridiculous. So people who are given marijuana as a prescription for glaucoma, there's actually so many medications out there that do a much better job of treating that pressure within the eye and reducing that pressure than something like cannabis. Speaking of which, glaucoma within the eye can also occur because of high blood pressure. So somebody who has just high blood pressure in general, they have high blood pressure because of genetic factors, poor diet, what have you. That can actually affect the blood pressure of the eye as well. So that's another source of glaucoma. Astigmatism is something that affects your vision. What's supposed to happen is the cornea is supposed to be a nice, basically perfect circle, perfect sphere. And then the lens should also be, equal in terms of its shape around the outside. It should be more like a baseball, not like a football. So if you imagine this ever so slightly not being a nice circle, not an actual football shape, but if the cornea or lens is slightly off in terms of its circularness, that can cause astigmatism, because the way that light is coming into the eye, it's not going to have the refraction of light as it should be. So some parts of light in your visual field, the angle at which they come in the back of the eye is going to be a little bit off because of that misshapenness. So astigmatism can be corrected by having the lenses that you wear adjust for that. Color blindness. It's more common in males. And if you've taken biology, you might remember that because of the sex chromosomes, that's one of the ways that red-green color blindness affects males more. Males have one X chromosome rather than two X chromosomes like a female does. And that can affect how the genotypes are inherited and being a carrier versus expressing it, etc. So color blindness, especially red-green color blindness, much more common in males. The inability to see color at all is extremely rare. That's one in thousands and thousands of people. It's very rare for your cones not to be, not having the ability to distinguish between all the colors of the spectrum. And if you've heard that dogs see in black and white, that's not true. Dogs probably don't see quite as much color as us, but think of it as more like closer to kind of like, that sepia setting on a camera that kind of gets to more browns and like different browns. I'm not saying that dogs see exactly like that, but if dogs don't see Roy G. Biv, they still have the different kinds of cones that allow them to see colors. Maybe it's not quite to the point that humans can see it. Speaking of which, like I said before, humans have 3 different cone varieties that allow us to see Roy G. Biv. There are some primates out there that have 4 cone varieties. So some of those monkeys could actually see more color than you and I can, which is amazing when you think about it. We might be looking at 2 forms of purple, and everyone agrees, oh, those are the same purple. But that monkey can actually distinguish between them, which is pretty cool to think about. Night blindness, one of the causes of it can actually be vitamin A. Remember, vitamin A is used to make that retinol that's inside of rhodopsin, which is inside of the rods and cones. It is recommended that you get vitamin A in your diet. But somebody who doesn't get enough vitamin A in their diet, you do have vitamin A reserves in your body, but once those reserves run out, you're going to notice that, especially at night, when there's less light coming into your eye, it's going to be hard to see certain things, especially while you're driving. So night blindness, one of the ways that you can actually cure it or fix it is get more vitamin A in that person's body so you can get those rhodopsin molecules to what they should be, how they should be functioning. A scotoma is a blind spot that's not normal. So if other than the one that's on the optic disc, someone else is having blind spots in their visual field, even with both eyes are open, that could be due to an injury. It could be due to lack of nourishment, then there could be genetic factors affecting a scotoma coming into play. Retinitis pigmentosa has to do with a dying off of those pigment discs inside of the rhodopsin. And that's something that could eventually make rods and cones just not function at all. So this is something that could lead to blindness over time. And a detached retina, this is something you don't want to have happening. A detached retina is usually from an injury. Some kind of injury to the head makes it so that the retina comes off of the back of the eye. And that means you're not going to be able to see. If that curtain against the back of the eye comes off, that's where all your rods and cones are. One of the ways that they can fix it, if they do it soon enough after the accident, is you can actually with a laser go in through the cornea and actually kind of solder on the sides of the retina on the back of the eye. You can get it back on, but the problem is when you laser on the sides of the retina, you are damaging those parts of the visual field. So that person's going to lose some of what the light that's coming in terms of perceiving it. But if it were me, I'd rather have some vision than no vision at all. Detached retinas, in other cases, the retina comes off and it's not fixed in time, and it's a permanent thing that causes permanent blindness. And like I mentioned before, one day we'll be able to remake a retina from stem cells. If you've ever dissected a cow eye or sheep eye, you know this, and if you haven't, I suggest that you try. The amazing thing is that when you cut into an eye and you look at the back where the retina is, it kind of looks like wet tissue paper. It looks very thin. It looks very fragile. And hundreds of years ago, when they first were cutting into the eyes and looking at them and trying to figure out what's what, they thought that the retina was just useless. They kind of tossed it like, that's useless tissue. Because they looked at this structure and thought it's not important. It's so tiny and so fragile. But arguably the retina is one of the most important tissues in the entire eye. And without it, you don't see. Thanks for watching Educator.com.